So it's very appropriate we're having a talk on microbe hunting. We have the micro hunter in chief giving that lecture, Ian Lipkin. Ian, I think it's not an exaggeration to say that Ian is the global leader in this field, has done more for the science of describing new pathogens than anyone else around today. In fact, Ian was the first person ever to describe a microbe using purely molecular means, born a virus back in the 1980s. It was a very big achievement, and not one that I think has been recognized for the magnitude, in fact, of what it was. A really, really key event. Today, he is the John Snow Professor of Epidemiology at Columbia University. He's got a fantastic lab up on 168th Street in New York. You can go there, lovely buildings by the bridge. And it's a great place to go, and they do fantastic work. So I'm very pleased that Ian's going to give our first talk. And Ian has agreed that tweeting is allowed, whatever that is. So during his talk, so tweet away, Ian.
Thanks, Eddie. I just want to say before I get started that yesterday, the past couple of days have been banner because we found out that there's a way out of the black hole. And I mean that literally and figuratively. So I'm not only referring to black holes, but also in addition, it looks like we're finally going to be able to get rid of Donald Trump because he put his foot in his mouth so many times at this point that I'm fairly confident that we're going to win. Okay, so obviously, autism to Zika is just a nice, convenient way to talk about A to Z. But I will, in fact, talk a little bit about autism and Zika, and I will be describing a number of pathogen discovery platforms and efforts that we've had that range from bacteria to viruses and parasites as well. And as Eddie said, I'm the John Snow Professor. That's an interesting story in its own right, but this is from the original cholera map that John Snow described in the 1800s, and you can see on the next one where the Broad Street pump was located, and he's popularly credited with stopping this particular outbreak by breaking the handle off of the pump. In fact, the outbreak was on the wane at that point, but it makes for very, very good figure indeed. And of course, what we now do is very different. We look around the world, and we try to find emerging infections, and of course, we use various sequencers. Most of us, my apologies to Oxford Nanopore, are still using the Illumina platform, but there are many platforms that are quite powerful. I'm going to talk with you primarily about acute diseases, but toward the end, I'll talk about where I think this field can go in thinking about chronic diseases. And here it's going to be quite important to think about the interaction of viruses, bacteria with the host genome, and the timing of the insult, which is also quite important, nutritional status, and a number of other issues. I got into this really serendipitously. I always wanted to be a clinician. And I started off in that route, and I went to do an internal medicine residency at University of Washington, and then neurology at UC San Francisco. And then we had this outbreak in these 5 young men of what was later proven to be HIV, leading to AIDS. And when it became clear that this was something that could be transmitted through the blood, this led this chase to try to identify the infectious agent. And it took 2 years from recognition of the clinical syndrome until the actual agent was identified, really at the Pasteur, not at the National Institutes of Health, but you all know that story quite well. And I was quite impressed with this, and I decided that we really had to come up with better ways to find and characterize infectious agents. So I went to work with Michael Oldstone and Floyd Bloom at the Scripps Research Institute to try to develop methods for understanding how HIV causes disease in the brain. And I was told quite confidently by Michael as I was beginning my fellowship that the vaccine for HIV was right around the corner. And we were instead going to work on lymphocytic corium meningitis virus. So in fact, as you know, we still don't have a vaccine for HIV, but at the very end, I hope to talk with you a little bit about the potential for vaccines for cofactors that may in fact reduce the risk of this disorder. Now, this particular work that Eddie was referring to, the identification of born disease virus, which took several years and involved enormous amounts of radioactivity and many, many molecular gymnastic techniques, can now be replicated with a wide range of platforms quite rapidly. That said, there are still new infectious agents that one can find that are not something that you'd be able to identify by simply doing a blast search. No matter how creative your bioinformaticians are, they will not find those agents. And I'll show you an example of those kinds of studies shortly. Now, more recently, we've tried to find ways to make it cheaper to do surveillance. This is not the only such system that's been developed, but it is quite powerful, and many, many people are now using it. We're trying to democratize access to it. I know Erasmus is now using it. There are a number of other people who are working with this as well. It's a very simple system. You simply make your library, and then you hybridize that library with oligonucleotides that represent the entire viral database, staggered at 100 to 200 nucleotide intervals across the genome, and then you do your pull down with magnetic beads, you sequence it, and it's really quite powerful. It gives you dramatic increase in capture rates, as you can see here, compared with the conventional methods we use, where we treat with nucleases, we try to eliminate ribosomal RNAs with things like these RNA binding compounds and so forth. And you can find in some instances examples where samples that have been tested by other people where nothing comes up whatsoever, in fact, wind up yielding interesting agents. And this was an example of a man who'd rejected an organ at the NIH. We were using this particular sample after we looked for a polyomavirus to see whether or not we could confirm it, and we could not find anything in the way of a polyomavirus. But in fact, when we finished characterizing, we found that he'd been infected with dengue when he'd gone back to Vietnam, and we subsequently found that infectious agent in the organ. So the method is literally as sensitive as real-time PCR. We're able to get it now down to 1 to 10 copies, and you get complete genomic sequence. The only thing you don't get is the termini. Now, as a result, in our lab alone over the past few years, we found hundreds of viruses. Most of these are not associated with disease. Eddie came to Columbia, I guess it was about a month ago, something like that, and showed me that he'd found something like an order of magnitude more. But in fact, the question is, how do you link these to disease? And we'll be talking about that in a moment. As you begin to use these types of approaches, it's very important that you be responsible. And this is, I'm going to be pointing the finger primarily at our community, but I'll also show you an example from bacterial discovery where we have the same problem. So we have the original finding, well, it wasn't really a finding, but it was a report in Lancet that MMR vaccine caused autism. We did a case control study. We thought we knocked it out. There was a subsequent study that we did looking at XMRV and PMLV in prostate cancer and in ME/CFS. And in each of these instances, the key was getting the people who reported the initial work to participate in the study and demonstrate that they couldn't find it. But every time you think this is dead, you become surprised. So we had this recent instance where Bob De Niro decided, after speaking with Andy Wakefield, that in fact the MMR vaccine was responsible for his kid's autism. And I managed to persuade him to pull his particular film from this very prominent film festival, and I thought we were done. And then a week later, he's right back on track with this notion that vaccines cause autism. So it's very important that as we do these kinds of studies where we find things using purely molecular methods, that we make certain that we clarify what's real and what we know and what we don't. Now, more recently, in the bacterial world, one of my colleagues at Cornell claimed that the New York subway had bubonic plague. Now, I took this quite personally because he looked at the subway station that I use on 103rd in Central Park West and said he'd found Yersinia pestis there. And what in fact he'd reported was by using very, very short fragments of the V4, V5 region, something that was in fact a Yersinia, but it was not Yersinia pestis. And this created an enormous amount of chaos. So again, pathogen discovery, pathogen de-discovery, it's sort of, the yin and the yang of this whole field that we now engage in. We're trying to focus now on developing other ways in which we can link infections to disease. So this means moving beyond next generation sequencing to what I like to call next generation sequencing, or something completely different. We began originally with peptide arrays, and I'm going to show you an example of how this works. These are collaborations that we've engaged in with Nimblegen. Nimblegen began to move away from oligonucleotide arrays, but they still had the capacity to print these high-density platforms. So they shifted to using peptides. Now, we looked at the entire vertebrate viral proteome, realized that we were going to need 24 12 residue peptides to tile that proteome, stepping by one residue all the way across. And the current feature density was unable to handle that. So what we then began to do was to winnow that down, 3 genes per virus, a tiling gap of 2, and so forth. And this method has actually proven to be quite useful. So in this example, we've looked at antibodies to MERS, antibodies to SARS, and you can see that discrete residues are recognized that allow you to discriminate immunoreactivity to these 2 different related viruses. You can do similar sorts of things with flaviviruses. Here we're looking at dengue and West Nile virus, and again, there are discrete epitopes. These are linear epitopes, but in fact, you can find that you can detect conformational epitopes as well, and I'll show an example of that shortly. So the National Institutes of Health recently gave us some support to try to build a serochip that would allow us to identify discriminant peptides that could be useful for differentiating infection with Zika virus and various dengue viruses. And this is quite challenging in this part of the world because dengue virus is endemic in Central and South America. And you'll be reading a paper shortly, not from our group, but from another, where Rafi Ahmed and colleagues have demonstrated that prior infection with dengue virus may have a role in aggravating the pathogenesis of Zika virus infection, possibly through antibody-dependent enhancement. So it's very important to identify the history of infection and understanding the pathogenesis and the vulnerability to these various reagents. Now, the problem, of course, in looking at this is that 73% of the antibodies to the Zika virus peptides using the approach that I've described, which is tiling the entire proteome, should have cross-reactivity with dengue viruses. Now, what happens when you print these peptides is that you begin to find that there are some that are clearly not specific, some that are cross-reactive, some that appear to be quite specific. This happens to be in NS1, and now we're in the process of testing this with larger numbers of samples, and this may allow us to move away from neutralization assays that are the current standard for differentiating these infections. And as you know, the neutralization assays aren't necessarily discriminatory. We ran into the same problem with West Nile virus and St. Louis encephalitis virus in 1999. All it means is that you have something that prevents virus entry or replication. Problem with the serochip that I've described for you is that it sells for $2,000. So this makes for very expensive assays. And we've been able to subdivide these chips into 12 wells, which gets us a little bit better purchasing power, but then we can't get the coverage that we want. So we've shifted to using phage display, primarily focusing on M13 bacteriophage and the GP3. And the approach is fairly straightforward. From the molecular vantage point, it becomes difficult when you begin to work with the vendors who have to produce the oligonucleotides. So what we've done is to take the same peptide library that we used to develop the serochip. So these are 36 nucleotide fragments that represent the open reading frames encoding those peptides. You have to make this particular cassette as small as possible because the longer the oligonucleotide, the more difficulties you run into with fidelity. You use PCR to add the adapters and then sequence the library. And this is an example of what I was talking about. So when we began working with the usual vendors, we found that there were all these gaps that changed or deleted or interrupted the open reading frame. So we shifted to very short oligonucleotides, used this company called Custom Array, And now we're able to generate oligonucleotides that give us the fidelity that's required. Because it's a big black box. You don't know until you get to the end of the production line and you begin using the library whether or not you've identified something that works. The system is quite simple. It's very similar to the one that was developed at Harvard by Stephen Elledge, but it's different in that we're using much shorter fragments. So we don't have the same sorts of problems that occurred there with cross-reactivity. You transform E. coli, you have your recombinant phages, you do your antibody binding, you collect those, they go into a sequencer, and it works very, very well. And we've piloted this initially, again, with flaviviruses. And you can see here now that what you do is you sequence these fragments, and this allows you, based on the quantitation of the presence of those products, to identify reactivity with West Nile virus and other things. Now, in some instances, you wind up picking up conformational determinants. So anytime two loops might come together in the final product, you may in fact bridge those. These tend to be somewhat lower in terms of the number of sequences that you obtain, but nonetheless, this illustrates that perhaps obviously with the exception of carbohydrates, you should be able to create libraries for a reasonable cost that will allow you to do this. Now, the oligonucleotides themselves cost about $250,000 to sequence, to synthesize. But once those are done, you can make these libraries ad infinitum or ad nauseum, depending on how you want to think of it. So I think this is going to be an excellent resource for anybody who wants to do this sort of work, and we're willing to share those, of course. I'm going to talk a little bit about new pathogen emergence, not that it's new to this audience, but to give you some hope that there may actually be some resources that will be very helpful in funding work that we would all like to do. And you know, of course, that there are many, many factors that go into the emergence of these zoonotic infections. This is a sort of a slide that I frequently show to politicians, particularly from the New York area, to illustrate the point of how we are at risk. We talk about the fact that we have 50 million tourists per year, 21 million international passengers, close to 200,000 flights, all the areas that you can reach nonstop. The green ones are flights that are coming into JFK. The red ones are the ones that are leaving. So clearly this is no different than London Heathrow or Frankfurt or any one of the number of large places that you might want to visit. For some reason, it's getting ahead of itself. Okay, so we also have this legal trade that we know about in pork, beef, and poultry that moves pathogens around the world. We have a large wildlife trade, which is larger than many of us anticipate. We have bushmeat, an enormous amount of bushmeat coming into Europe, for example. China, we really don't know what's going on here. We estimate it's less than 100,000 kilograms coming into the US, but nonetheless, so we've begun looking at those bushmeat samples that come in through JFK Airport, and we have found retroviruses and herpes viruses and so forth. Now, in the past, the majority of these samples that come in, such as these chimpanzee heads and so forth, and this is a monkey head, are desiccated or smoked. But we're beginning to see fresh meat coming in as well, so this is going to be an enormous challenge. One of the questions that frequently comes up is how much uncharted territory is there? And we've tried to estimate global viral diversity by using a number of laboratories around the world where we get some insights. And I'm just going to show you one example of where we've tried to estimate the zoonotic pool by looking at flying foxes in Bangladesh, living in close proximity to humans. And this, just using consensus PCR, we were able to identify 55 different viruses, 50 of them novel, or according to ICTV-DB criteria, and then estimated, based upon the recovery of new sequences, using this sort of an approach, and thinking in terms of the number of mammalian species, which is roughly 5,500, that there were a minimum of 320,000 viruses that could be captured by using consensus PCR. The number, of course, is going to be much larger if you begin to use high throughput sequencing techniques. We then extrapolated this cost for characterization of all of these in 1 point to $6.3 billion, $1.4 billion for 85% coverage. And I actually had a call from the World Bank that wanted to see whether or not this was something feasible that they might want to do. And so I gave them the cost, and they talked about the cost of SARS and H5N1 and various other viruses, and they were initially interested, but nobody ever followed up. And then just a few days ago, I got a call from USAID. Peter Dajak, who some of you may know, is organizing a meeting in New York with USAID on the 20th of this month. And then there's going to be a meeting in the Bellagio, and they're going to try to actually generate the resources that would be required to pay for this kind of work. And if, in fact, this comes to pass, there's going to be enough work for everybody in this room, because I don't think any one lab can handle all of this sequencing. So wish us luck. And if anybody asks you whether or not a global virome project is worth doing, the answer, of course, is an enthusiastic yes, right? Okay. Now, I thought I would tell you a little bit about this, although everybody thinks they know all about the story of MERS and so on, but to illustrate the points that very few people, except perhaps Marianne from Erasmus, may know of what it's actually like to go out and do a field trip expedition and the things that can go wrong. So when MERS was originally appeared in public consciousness, there was this one man who was 60 years of age from a small town in Saudi Arabia known as Bisha. And samples from his respiratory tract were used to isolate a beta coronavirus by someone named Zaki, who's now no longer in Saudi Arabia. It was sent for sequencing at Erasmus, and Ron Fouché identified this novel coronavirus. And then the question came up, where did it come from? So I was contacted by Ziad Memesh, who was at that point the Deputy Minister for Public Health, and asked to see if we could identify this infectious agent. But what we found very rapidly was that nobody had done even the most straightforward epidemiological investigations. So I'm going to illustrate that point for you in a minute. So we flew to Bisha via Riyadh, and this gives you an idea of where Bisha is. It truly is in the middle of nowhere. We went to this man's house. He had 3 houses. He was in the process of building his 4th for his 4th wife until he died. And when I tried to get some sense as to whether or not he had any chronic illnesses, I was told no, and obviously the proof was that he was about to take a 4th wife. And I said, I've had difficulty with one spouse, and I know most women claim the same about men, so I'm probably on safe grounds. So this was his son. This is John Epstein from EcoHealth, and 2 Nigerian veterinarians who are doing some of this work. And we met with a number of people. This is his brother. Nobody had actually spoken to him about how he might have contracted the disease or how he was sick or anything else. Subsequently, I went and participated in some analyses of what was happening with hospital transmission. We learned chiefly through McKinsey & Company, which is a business consulting firm. I was given access to the hospital, and I found that people were using N95 masks over full beards, so they weren't even getting a seal. And because of various concerns about the cost of insurance, people were being placed for dialysis or treatment or whatever in emergency rooms, literally gurney next to gurney. So if anyone was sick, everyone got sick. So it was very interesting to see how this got put together. So we began looking for things, and we'd recently found that there were some bats that had related sequences. We were told by the Ministry of Health that there were no bats in Saudi Arabia, and we said we wanted to look at bats, and we were interested in camels. So we brought this back. This was from this man's place of work, which was immediately behind the garden where all these bats were flying around. And this was us swabbing camels. One of the problems in that first expedition was that we were unable to bring any camel materials back to the United States because of concerns about foot and mouth disease. So the initial link actually between camels as the most important reservoir was made at Erasmus, rather than in New York. And this is our driver, and here we're getting ready to go look for a variety of things. So we found this one Tophosis bat in this area very close to where this man had his place of work, and we found identical sequence. We've not done any extensive bat studies since, but we presume the origin of this virus was a bat to a camel. Camels became the amplifying host, and that now is the most important animal model vector. Now, in order to do the work in Saudi Arabia, because it became increasingly difficult to move things back and forth, and the US and the Department of Agriculture didn't have materials, we began creating these mobile labs where we would have extraction machines, materials for serology, PCR, and so forth. And these two guys went off and wound up characterizing these. And we found, not surprisingly, that the majority of the adult camels in Saudi Arabia in the survey had evidence of infection with this virus, but they did not have quite as high evidence of active infection. So 35% of the juveniles had nucleic acids, and we recovered virus there as well. So presumably, and many of you know these data at least as well as or better than I do, this again is the evidence for that particular reservoir. But there was enormous resistance to the notion that camels could be the reservoir for this virus, despite the fact that there was work coming out of Erasmus and Sanger and lots of other places. And so we began showing these pictures from Riyadh of wild of camels, close proximity to the meat. Here's camels being let out to drive. This is just on the outskirts of Riyadh, and this is people becoming very, very intimate with camels indeed, right? They do look like testicles, but in fact, they're just swollen tonsils. And finally, now we have an agreement with the Department of Agriculture where these people are being trained so that they can actually do that work and not send materials out of the country. Now, I pointed the finger at the developing world, but we've got plenty of uncharted territory in New York. So this is between Chinatown and City Hall, roughly noon. And you can see all these rats wandering around, and they're obviously doing quite well. And I like movies, so this is from Gangs of New York, if any of you have seen this. So we call this paper Rats of New York, and this is the same sort of picture of Daniel Day-Lewis sort of looking rat-like off into the distance. And what Cayley found was that there are a wide range of bacterial pathogens that were present in these rats. I'm not saying that they're necessarily resulting in human disease, but in this instance, we sequenced them from stem to stern, and they truly are there. Most interesting thing we found, however, was a Hepatovirus, which has now become the model that people are using for studying human Hepatoviruses now that they no longer have access to chimpanzees. So as you do this sort of wildlife work and rat work and so forth, you can find a lot of interesting viruses that have utility beyond identification of human disease. In addition, we've begun looking at ticks, and we found there are a number of people, I'm sure you have them here in the UK and Europe as well, where people are treated with antibiotics and they don't respond. And they develop this syndrome known as chronic Lyme disease. So our hypothesis now is that these people are infected with viruses. This is a tick. This is a tick that's had a blood meal, and they sit attached to you for very long periods of time. And you can find that they're infected with a wide range of bacterial pathogens, but they also have interesting viruses. So here's an example, paramyxoviruses that are interesting. They're rhabdoviruses. And the question, of course, is whether or not people who have these chronic diseases have antibodies indicating that they've been infected with this virus. So this is the paradigm that we now use. And what we found is that you can rapidly identify seroreactive individuals. Here's an example using a method called LIPS that I don't have an opportunity to go into, but these are all individuals with quote-unquote chronic Lyme disease who did not respond to antibiotics, who have antibodies to this virus. And the question now is whether or not this virus is responsible for their symptoms. So this is the sort of approach that one can take, and by using these peptide chips, you can finally discriminate reactivity to viruses, bacteria, and so on. Now,
I give your majesty the gate of martial valor.
So this is sort of the beginning of microbiome research in our lab. And as you see, these guys are very excited to look at these feces rolling around. And nobody in my lab is ever this excited about their data. But we're hoping that this is going to change with this new bowl to biome project, which is imminent. So we've actually been working in this field for about 5 years, and we have published very little. A couple of papers on autism and so forth, but that's about to change because there's some really cool stuff that one can find. And others are beginning to make the shift a little bit. Eddie's also doing some bacterial discovery. It's a lot of fun. And it's no more difficult than doing the virology. So, and if you think about the fact that our DNA is largely common, we have an enormous amount of diversity in the microflora. And these are very important, not only in digestion, but in tuning the immune system and influencing behavior and having an impact on vaccine uptake and so forth. And it potentially implicated in a wide range of disorders. Now, modern living has changed our microbiota. Again, I'm just running through this very quickly, but I think this is something that virologists need to begin to think about too, because there are interactions between bacteria and viruses that are extremely important. The only current approved use in the US for fecal microbiota transplantation is for Clostridium difficile. But people are beginning to use it for a wide range of purposes, for Crohn's disease, ulcerative colitis, irritable bowel syndrome, even variety of autoimmune and allergic disorders. And we have some data now suggesting that there are certain types of colon cancer with an abnormal methylation phenotype where the microbiota differ. And they don't produce butyrate, and butyrate is important because it actually arrests the cell cycle and results in apoptosis. And you get more aggressive tumors if, in fact, you have the wrong sorts of microbiota. So I'm going to give you a couple of examples of how this works. This is a disorder known as stillbirth. It's more common in the developing world than it is in the developed world. But nonetheless, it's still extremely important. So what we decided to do was to look at placentas of individuals who are live-born or stillborn. First of all, to measure the quantities of bacteria that are present in those organs. And we found that case placentas, that is to say stillborn placentas, have far more bacteria than control placentas. And we found the same bacteria in the case livers, the stillborn livers, that we found in the placenta. So some didn't make it all the way across, but we didn't find any examples where there was something present in liver that was not present in placenta. So with that feasibility in hand, we then decided to characterize the microflora of these placentas. And we found that there were different biomarkers, different dominant bacteria in those kids who were live-born versus the ones who, in fact, were stillborn. And you can see that. So the green ones are the live-born, the red ones are stillborn. And we found that there were differences in levels of bacteria and the types of bacteria that were present. If you looked at children who were stillborn at less than 23 weeks of age versus those who were born after 23 weeks of age. And when you look at the bacteria that are most important, the one that really jumps out is Streptococcus agalactiae. So we decided to focus on that because it's extremely important as a biomarker for stillbirth, and it's particularly important in that we vaccinate against this particular bacterium because it's a common cause of disease and actually even death in children who were born, who passed through a birth canal where there's an infection with Strep agalactiae. But in fact, women are screened for the presence of this bacterium at 35 to 37 weeks gestation. A trivalent vaccine is given to some at 28 to 36 weeks gestation, but the vast majority of disease is present before 23 weeks. We then had to serotype these bacteria because you cannot use molecular methods to do this. So we had to then develop antibody tests to serotype all of these bacteria. We found that the vaccine didn't cover two major serotypes, 2 and 5, and because it was given too late, where you missed 33% of the stillbirths, and you missed 2 of the major serotypes, as you can see just by summing these, you missed 3/4 of the vaccine-preventable stillbirth, which is extraordinary. So these kinds of findings can have a huge impact on public health. And I think they're important not only in terms of virology, but to think about this as microbiologists as well. Now, I wish I could talk more about this, but I can't because there's going to be big sort of release around the discovery. But I'll show you what we've been doing. So as I mentioned, when I began working with Mike Oldstone in 1984, I wanted to work on HIV. So we sort of come full circle, and this came about because we were interested in the observation that there were a group of women aged 15 to 24 in sub-Saharan Africa, primarily in South Africa, in and around Durban and KwaZulu-Natal, who had the highest risk of heterosexual infection with HIV/AIDS. And Salim Abdul Karim and his partner Carisha identified these women and began following them, collecting vaginal washes and such, and found that there was an extraordinary relationship between the presence of a set of 4 pro-inflammatory cytokines and risk. So that if you follow these women long term, you found that those who had high levels of these cytokines wound up being at dramatically increased risk for subsequent diagnosis with HIV and progression to AIDS. We reasoned that there would be something that would be driving this, which would be microbial, so we began looking for bacteria, fungi, and viruses. And I can tell you that it's not a virus, and it's not a parasite. It is a bacterium. And if you're really quick, you can see which one it was there. But now I think we can actually get at the idea that there's a bacterium, a single bacterium, which appears to be the major driver, not only of inflammation, presumably drawing susceptible cells down to the vaginal fault, but actually having an impact on the mucosal barrier. And we can vaccinate against this bacteria, and we may have an impact. Shifting away from acute diseases to chronic diseases, I'm going to share with you some extremely cool data on autism now. So when I began working with Michael, shifting and thinking about terms of lymphocytic choriomeningitis virus, we began to focus on the notion that he and a neuroscientist with whom he was working, that there would be a single virus for every neurotransmitter system and a single neurologic disease for every sort of system. And of course, it proved not to be as simple as that. And I became increasingly impressed by the fact that there was some sort of a non-specific innate immune response that resulted in damage, behavioral disturbances in offspring of mice and rats and other animals. And one could use lipopolysaccharide to mimic gram-negative bacteria or double-stranded RNA to mimic replicating viruses and get the same effect as you would get with influenza viruses. Paul Patterson, who died a couple of years ago, demonstrated this first by using H1N1 influenza, and he found that if you exposed an animal roughly halfway through gestation and you looked at an activity track plot, the animal that saw the virus was huddled in the corner of a cage, whereas the control animal ran around and explored his environment. And if you administered the same stimulus, gestational day 16, so roughly 3/4 of the way through gestation, instead of being withdrawn and huddled in the corner of a cage, the animal was hyperactive compared to the control. Very interesting. Same genetic background, same exposure, different timing, profoundly different effect. We subsequently demonstrated that you're knocking out specific populations of neural stem cells depending on when you expose the animal to that particular infection. But the question is, how do you then link this to human disease? In 1999, shortly after we discovered West Nile virus, I began talking with people at Columbia about moving there. And what was most attractive to me was the ability to get into the ground floor of a birth cohort that had been established in 1999 out of the Norwegian Institute of Public Health. And what was interesting about this was that we had the opportunity to define the study design, the questionnaires that would be obtained, and the samples that would be collected, so that as children would progress through life, we could look at gestational determinants of health and disease. What we have found so far, and these are unpublished data that are very cool, is that if a woman has more than 3 episodes of fever after the 12th week of pregnancy, she has a dramatic increased risk of having a child who's ultimately going to receive a diagnosis of autism. So this actually is the epidemiological correlate to what I showed you with the mouse studies. We've also been able to look at the impact of paracetamol, as it's called in Europe, or acetaminophen in the US, on the risk if you have these episodes of fever, and we found that reducing fever actually has a protective effect. But if you take acetaminophen for any reason other than fever, it results in an increased risk. But again, it's a sort of a dose response, which makes sense because you're knocking out specific cells and circuits. We've also begun looking at cord blood from these children and have found that the children with autism have elevated levels and decreased levels of different cytokines and chemokines. So this allows us now to begin to think about biomarkers, risk factors, all presumably related to infectious diseases. Now, what infectious diseases might those be? So we're doing some work right now with influenza, but it's taking a long time because we've had to look at something like 19 different viruses, all with hemagglutinin inhibition assays, which is an enormous amount of work when you're talking about close to 500 subjects and all the controls. But we have been able to complete torch assays, and the only thing that's come out of that has been that we can rule out toxo, rubella, cytomegalovirus, herpes simplex type 1, but it appears as though an infection with herpes simplex type 2 may be important. This is not surprising if you think about it. It's an infection in close proximity to the placenta where it might in fact have an impact on the integrity of the mucosa or induction of innate immunity. All of that remains to be worked out. But this is the first sort of solid evidence I've shown you, the history of fever, herpes simplex, cytokines, chemokines, to suggest that this disorder, which many people have thought of as being purely genetic, may not be the case. So there's lots of opportunity to do interesting things. I'm going to talk with you about this virus not because it's going to have a huge impact here, but because it's only the second virus in my entire career that we would not have found by comparing sequences to a database. So this is a virus that infects tilapia. It's not a particularly tasty fish. I don't know if anybody likes tilapia here. We initiated this project simultaneously with a group in Ecuador and a group in Israel. The Israeli group actually gave us better samples, and that's where we ultimately found the virus. It is an important virus in the developing world. People eat a lot of it, and in fact, it does have some utility in controlling breeding mosquito populations and larvae and so forth. And these are all the areas where you have tilapia farms worldwide, and you can see these are dead tilapia. This is a tilapia with a distended liver, and again, we began looking for this in Israel and Ecuador. What we found, in fact, was that this is a virus with 10 genome segments. When we looked at the initial sequences that we obtained, we found terminal sequences that were homologous to one another, which suggested that they were viral. There were 10 that didn't match any sort of fish database, and that there was a remote similarity to the PB1 of influenza C in the largest of these gene segments. But we're talking about 30% at the protein level. So it's not something that would jump out at you and say, wow, this is clearly a new orthomyxovirus. And this correlated then with the presence of these capsids that were identified by EM and in situ hybridization, demonstrating the presence of this. So this particular virus, we've now been able to demonstrate, in fact, that we can use these individual probes representing those 10 genomes to demonstrate the presence in northern hybridizations, that there are in fact products. And the most difficult part of this whole study was resurrecting northern blots. Who here still runs northern blots? Anybody? One hand went up. So I was, taught to run northern blots somewhere back in the prehistoric era. It's very, they're very, very important. When you find new viruses, it's really the only way to sort out what's relevant. Now I'm just going to close by showing you one example of a non-infectious disease. This is actually work from a long time ago, but when somebody shows you something that doesn't make sense, I want to encourage you to pursue it nonetheless. There was a processing plant for pigs in Austin, Minnesota, where various workers were beginning to fail on the assembly line. They lost their ability to manipulate their fingers. They were unable to walk normally. They all began presenting at the Minnesota Department of Public Health and at the Mayo Clinic. And I was contacted by someone who wanted me to look for a prion disease using a gene chip that we had recently modified and adapted and so forth. And I explained to these people that, in fact, there was a problem with detecting prion diseases using a gene chip. But I wanted to hear more about the story anyway. And what we found when we sent people out there to look at this was that this processing plant took 30 pigs through every single day. And the pigs would come into one side of the plant. Every place you see a blue rectangle is where there's a worker on an assembly line who's taking off some piece of the pig. And the nice bits of the pig are here, if there are nice bits of the pig. And the yellow rectangles refer to areas where somebody's removing the brain or packing the brain. Now the question, of course, is what would you do with the pig brain? Well, there was an enormous amount of interest in these pig brains in China and Korea. And the question was, how are you going to process 30,000 pig brains? So a rocket scientist who joined their staff had come up with this idea of taking a high-pressure air hose, running it through the base of the skull of the pig, liquefying the brain material, and having it run down like so. So this is the snout of the pig. Here's the eye. There's a pressure plate here. I think you can see an air hose. Does that project? There's an air hose. And you can see that this individual is not really wearing much in the way of PPE. Wasn't wearing any sort of a mask, for example. So when we began looking at serum from these individuals, we found that they had antibodies to pig myelin, which is the insulating substance in the peripheral and central nervous system, and that those antibodies to pig myelin cross-reacted with human myelin and resulted in an autoimmune disease. So this particular project, which we ultimately reported in 2010, which started as pathogen discovery, wound up becoming something very, very different. And the solution here was to not use this air hose anymore. So it was the world's cheapest vaccine, if you will. Now, obviously, I don't do the majority of the work that I've shown you here. Very, very talented group of people, and I've tried to show you pictures as we went up, but I wanted to allow enough time for questions. And I thank you very much, and I really appreciate the opportunity to come and hear what you all have to say over the next few days. Thank you.
Well, that was fantastic. I'm allowed to chair the question session, and there's some microphone runners, so please use them. So questions for you?
So you showed a toilet with a sequence on it, and I only saw patent pending, but nothing other than that. That's actually an idea that we've talked about as well. So I'm just wondering, what are your plans there? Because it seems pretty disgusting, and then you think about it, and you're like, that's brilliant. So what are you guys actually doing on that?
So the question really is this notion of analyzing fecal material in a very rapid way. How are we going to proceed with that? So when we initially began working with 454, which gave us very long segments for V1, V3, well, for those of you who don't do this kind of work, there are really two major strategies. One is you look at specific regions in the ribosomal RNA that you amplify and sequence, and the other is you do complete genomic sequencing, metagenomic sequencing as it's called. And obviously metagenomic sequencing has major advantages because you can get insights into pathways, products that might be made by bacteria, and then you can look in plasma or spinal fluid to see whether or not those particular products are present and link them back to the original bacteria, which really gives you a more complete picture of pathobiology. So we began with 454, and we still do, believe it or not, we still do some 454 for this purpose. At the V1, V3 region, when people began moving to Illumina and getting shorter reads, they began using V4, V5. And the V4, V5 in our hands has proven to be quite misleading. So what we've now done is we've come up with alternative primers for V1, V3, which are very useful. They're not yet published, but if you come up and find me, I'll put you in touch with people who are now using those. Those, I think, are still the best way to do surveys because metagenomic sequencing is still too expensive. But we're using this now to look at colon cancer, inflammatory bowel disease, the vaginal microbiome, chronic fatigue syndrome, and a number of other disorders where we think that there may be triggers. We focus on feces because they're so easy to obtain, but we're also beginning to look at saliva. One of the challenges is that different regions within the same organ may have a big impact. So the work we originally did with autism, the mucosal microbiome is different than what is present in free fecal material. The gingival crevice in the mouth is different than the posterior oropharynx. The cervical region is difficult than the anterior vaginal fornix. So it's very important that you use standardized methods when we're comparing these so we can actually begin to understand what the microbiome really is.
But are you actually developing that as a toilet, though? Because it's one...
No, I'm not developing that as a toilet. I think that's something we could approach. We could approach Toto on that. I think they might want to add that particular feature. But you know, I remember the first time I was doing a sabbatical in Germany. I was in Würzburg, and there was this shelf in the middle of the toilet. And I asked people what that was for. And they said, well, that's so that you can examine your feces, should be interested. So apparently they still make toilets that way. I guess we could extract them. Why not? You're shaking your head. Somebody's shaking his head. No. That's no longer the case? Anyway.
Oh, here. So from your talk, it looks like all the virus has linked with most of the diseases. So as I wonder, so far all these viruses are selected from bats. They are all from outside the U.S. As far as I know, you also have bats. Especially I remember that when I was in Austin, a lot of bats under a bridge. Have you ever tried to see, whether you have some viruses in the American bats? Thanks.
So George Gao is asking a very insightful question. So if we don't look for, and his question has to do with whether or not anybody has looked at American bats. There is, I know Ralph Barrick has looked at bats from the bridge underneath, in Austin, which is where they frequently have American Society for Virology meetings, but I've never seen any very interesting reports there. Most people are looking for things where they think these emerging infectious diseases arise. Closer to home, we're focusing on rats and mice and ticks.
So a feature of many of the examples you showed is close attention to proving cause rather than association. But as you proliferate the number of viruses and bacteria and microbiome determinants that you assess, how do you think we can mitigate in the future against false positive associations, etiological associations, when you test so many different things?
So the question is, how do you minimize the risk of artifact which can result in people implicating agents that that are basically not relevant? It's a very, very good question. So a couple of years ago, I published an editorial in Nature Reviews of Microbiology where I talk about levels of confidence in the relationship of a finding, because clearly we can't go through all of Koch's postulates at this point. What we do is we look for localization through in situ hybridization, because we don't want to invest in making antibodies until such time as there's a reason to do it. We look for high levels of the infectious agent as being present. We actually go back to the work that Thomas Rivers did during the original 1918 flu pandemic by finding antibodies as an indication of the fact that the virus actually crossed over from an animal into human and resulted in an active infection with expression of an immunogen. But at the end of the day, if you can't develop an animal model that allows you to rigorously test that, you have to look for biological plausibility. So If it's an acute disease, this is relatively straightforward to do because you have an example in another animal species where a virus is replicating, causing disease, and that may be sufficient to prompt somebody to develop a vaccine. And this is what happened with papillomaviruses, right? And now, of course, we have a human papillomavirus vaccine, which has reduced the incidence of papillomavirus infections. And similar things are probably going to happen here. With respect to the bacterial world, there's something which I didn't mention, which I think is extremely important. When you're looking at very rare sequences, let's say, for example, you're examining air filters or relatively sterile fluids like spinal fluid, sometimes there can be material that carries forward with the reagents you use for PCR and for sequencing that wind up becoming sequenced. So we've come up with ways in which we can take buffers and enzymes and treat them with 4-base cutters and eliminate the template that might otherwise get amplified. Short of that, I don't have any other tips on how to reduce this except that make sure somebody can reproduce your work. I will say this, there's a very short lifespan or memory span for people who screw up. So you can implicate all kinds of things, walk away from it and get away with it apparently. And I can give you some examples of that over a beer.
There's one at the back then.
Marion?
That's one first then, Marion.
One of the big issues is the contamination that you get with platforms. How do you deal with that?
With which now? With viral discovery or with bacteria? So with bacteria, as I said, we digest with 4-base cutters everything we can. We treat everything else with UV. So for reducing the propensity for contamination, which is a huge problem, we have special rooms which are outfitted with ultraviolet light, and we assemble the PCR components, with the exception of the sample, in a positive pressure room. So again, it sort of reduces the risk of people coming in. Other than that, all you can do is have anterooms where people come in and change. If it's introduced at the level of the sample, that's a larger problem. But the one thing I would say is I never accept a sample from somebody who has found something where they think they have found something novel and they want me to confirm it. Because what I've always found is, yes, I can confirm it, and then they want me to sign on as an author on a paper, and it may or may not be true. So what I prefer to do instead is to set up a blinded analysis where we collect the clinical samples and we send it to you under code, and we look for consistency when you test the same sample more than once. That's the way we did MMR and autism, the way we did XMRV and chronic fatigue syndrome, enteroviruses and ALS, and so on. The real challenge that nobody's asked about yet, which is surprising to me, is how we're going to get insights into some of these things that cause disease by hit-and-run mechanisms. And that's why I sort of ended that last slide on an example of autoimmunity. So we still don't have a good way to look for these sorts of events. And it's going to require the same sort of investment in serology and in host response that we've made in genetics, which is frankly becoming pretty simple to do because it's being done everywhere.
There's one last question.
I would like to ask a question about the protein array and phage display assays that you showed at the start of the talk.
Oh, there it is, yeah.
You mentioned that these were detecting antibodies that bound to conformational epitopes and said that they might be a good alternative to neutralization assays. I was interested to know if you had a handle or some idea about what proportion of antibodies binding conformational epitopes they detected, seeing as you're only using peptides, and as an extension of that, what proportion of antibodies which bind conformations which you're not detecting, as we know that most antibodies bind conformational epitopes.
So the question is, what insights do we have from work with the peptide arrays into the relative proportion of conformational epitopes that are reactive with these sorts of antibodies? The answer there is that we don't know. So what we did is when we found that there were these reactions that came up with sequences that were obtained in viruses that shouldn't have come up, we then went back and looked at structural models and found that there were adjacent loops that explained the finding. And that then gave us hope that if, in fact, there is a structural model available that, for a related virus, you can use that then to add to this particular array by simply synthesizing additional peptides that would represent the predicted conformational epitopes. And once that is done, it should be possible to directly address the question that you've asked. We don't have that as yet.